Thank you, Matthew. Our next speaker is Emily Behrman.
Hi, so today I'm going to present a piece of the research that I've been doing during my dissertation, which is looking at life history adaptation in Drosophila. My talk is going to address two fundamental questions about the process of adaptation in natural populations. First, what is the rate at which populations respond? Then to the changing environment. And then I will address the genetic architecture underlying the translation from genotype to phenotype and how that affects the timescale at which populations respond. Evolution is traditionally considered to be a very slow, gradual process of small incremental change over a very long timescale. So it's thought to be something that we can infer, but not necessarily measure in real time. There are instances which demonstrate that populations do respond adaptively much faster than traditionally assumed. And this has sparked this dialogue about the idea of evolutionary processes occurring on ecological timescales. This is also known as rapid adaptation. We still lack a fundamental understanding on just how fast populations can respond and the genetic processes that underlie this rapid phenotypic change. So today I'm going to address this using natural populations of a genetic model. I'm going to look at selection across time. Seasonal rhythms produce predictable changes in the environment over a rapid timescale. This means that for arthropods, such as Drosophila, as well as other multivoltine organisms that reproduce multiple times each year, each generation is experiencing drastically different environment. And my question is if and how these populations respond to the environment and what this can tell us about the process of adaptation. I'm making predictions about selection across time based on well-documented examples of selection across space. By looking at parallel latitudinal clines worldwide, we have produced the generalization that temperate environments select for an elevated stress resistance, whereas tropical environments select for an increase in reproductive output. This encapsulates this classic life history trade-off of allocating limited resources between reproduction and survival. And so I'm predicting that similar environments will select for similar traits both across latitudinal space and in a single location across seasonal time. There's historical precedence for this parallelism. Dobzhansky observed that chromosomal inversion frequencies in Drosophila pseudoobscura cycle in the same way across seasonal time as they vary across space. So I'm specifically predicting that a temperate winter will select for elevated stress resistance, and populations that are sampled in the spring directly after that will have a high stress response. Due to known fitness correlates and trade-offs throughout the summer, I expect that the stress resistance will decline and therefore be low when sampled in populations in the fall. But because the seasons continue to cycle, I predict that stress resistance will then increase again in the winter as a correlate of overwintering survival. So if populations can respond to this rapid seasonal change, we predict that the stress traits will oscillate over seasonal time. I measured this by collecting flies across seasonal time and from their natural environment, which is fruit orchards. I established isofemale lines and maintained them in a common laboratory environment, which should remove any environmental or maternal effects. Therefore, when I measured traits, any difference should reflect a genetic basis of change in the population. I measured a panel of fitness-associated traits that are known to vary across latitudinal clines. And what I show here is the trait values on the y-axis for all of the traits measured are changing in a rapid way across seasonal time, which is shown on the x-axis. And this is happening at a very rapid speed. It's an estimated 10 to 15 generations between the spring and fall endpoints. Additionally, for all the traits measured, the pattern of change is matching the prediction of elevated stress resistance in the spring and a decline throughout the summer. So flies survive longer, under starvation conditions and heat knockdown. They recover faster from a chill stress, and they develop faster. And so we're seeing that overall, across this very rapid timescale, there is both rapid and pronounced seasonal change, and it matches the predictions. So the question is, how representative is this? Is this repeatable across time and space? I've just completed the 5th year of my PhD, and I now have a 5-year longitudinal data set on the same population. You can see that there are repeatable oscillations in all of these traits. There's some secular variation from year to year, presumably due to the differences in the severity of the environment experienced across time. But overall, for these complex quantitative traits, we're seeing a repeatable, predictable pattern across seasonal time, which is demonstrating a genetic change in these populations on a rapid timescale. To further investigate this, I maintained all of the isofemale lines in the lab until the collections were complete, and then I reconstructed the populations by allowing the lines from each collection to intermix so I could simultaneously measure this longitudinal sample and see if the patterns persisted. So these plots are oriented in the same way with trait value on the y-axis and seasonal time on the x-axis. And you can see that there is a profound difference between spring and fall populations that is maintained even in the absence of seasonally based selection. Additionally, I looked at the variance-covariance matrices across these populations and found that there are distinct differences in the G matrices that are repeatable across seasonal time. So this is showing the genetic basis of the populations are changing rapidly and repeatedly. To look at this seasonal change in the context of the well-documented clinal variation, I sampled simultaneously across space and time. And I followed the same collection regime that I previously described. So this is the same figure that I previously showed you of the wild isofemale lines, but I've added the Massachusetts collection in blue and the Virginia collection in red. And so there are 2 points I want to emphasize here. The first is that selection across seasonal time is incredibly parallel across these collections, across these locations, that they are all responding in a strong way to seasonal change. The second point I want to emphasize is that the magnitude of change from spring to fall is much greater than the difference experienced across these spatial scale. In fact, when we look at the magnitude of change in any of these populations from spring to fall, which is again approximately 10 to 15 generations, the magnitude of phenotypic difference is equivalent to the difference that's been previously measured in populations of flies from Maine to Florida, which is spanning 20 degrees latitude. So overall, I think I've very thoroughly demonstrated that populations can and do respond very rapidly to the changing environment in a strong way. And so now I'm going to move from looking at these natural populations and take advantage of the genetic tools and resources of this model organism to understand the underlying genetic architecture. So with my collaborators, we did whole genome resequencing on these same populations and saw that across seasonal time, which is on the x-axis, the allele frequencies on the y-axis of hundreds of SNPs cycle in a predictable and repeatable manner across seasonal time. This brings up 2 questions. First, do these SNPs have a function to affect the phenotypes that we observed are changing? And second, do they act in an additive way, or are there epistatic interactions that help facilitate this rapid response? So to address this, I'm going to look within the gene couch potato. A single SNP within couch potato has been previously mapped to be involved in overwintering diapause. The frequency of the high diapause allele increases with latitude, predictably, it also occurs more commonly in the spring than the fall. So I use whole genome resequencing across a latitudinal cline to identify additional SNPs within couch potato. So the position within couch potato is on the x-axis, and the degree of clinality is on the y-axis. I'm going to focus on these 3 SNPs. This blue one is the one that was previously mapped, and there's 2 other SNPs that are highly clinal. All 3 of these are independently clinal, and they also 2 of them cycle with season. The SNPs span 3 kb, and pairwise linkage disequilibrium between the SNPs is low. All of the different haplotype combinations occur in the wild, and so all of this together suggests that these SNPs are acting independently. And I'm going to use this as a microcosm to test if these apparently independent SNPs are acting together in a non-additive way. I'm going to focus on looking at the temperate combination compared to the tropical combination. So to do this, I created population cages that are consistent for each of the different haplotypes using a panel of inbred sequenced lines. And I have to say that this one combination occurred at too low frequency, so I could not create this one haplotype. So the cages were created by taking lines for each of the different haplotypes and allowing them to recombine. Therefore, I ended up with, for each haplotype, I ended up with a population that was fixed for the alleles of interest, but had a heterogeneous background. I then tested these populations for a panel of fitness-associated traits, and I created a null model of additivity based on the individual alleles that compose the haplotypes. You can see here that the temperate and tropical haplotypes are not predicted to have very strong phenotypic differences. When I measured these haplotypes, however, I find that there are strong non-additive effects of the alleles, specifically that the temperate haplotype has a much higher overwintering diapause, starvation resistance, and body size, which are all traits that are known to be involved in temperate adaptation. In contrast, the tropical haplotype has much higher reproductive output than predicted in the additive model. This demonstrates how interactions among a few SNPs can produce these emergent phenotypes that are involved in environmental adaptation. And this can help facilitate the rate at which populations respond. Additionally, we know from previous work that decreased level of couch potato expression is involved in this suite of fitness traits associated with temperate adaptation. And I see that there is non-additive decrease in couch potato expression within this temperate haplotype. And so this is probably driving these emergent fitness properties. So I began by posing 2 questions about the process of adaptation. First, how fast are populations changing? I have demonstrated that the rate is rapid and the pattern is repeatable. Natural populations of Drosophila melanogaster evolve in response to seasonal environmental changes for suites of fitness-associated traits. The magnitude of change we're seeing across 10 to 15 generations is equivalent to documented differences across 20 degrees latitude in North America. Next, I asked about the genetic processes underlying these traits. We see that there are hundreds of SNPs that cycle with season, and I've shown that at least in the case of couch potato, this is driven by the SNPs are functional, and there are epistatic interactions that produce emergent phenotypes that are involved that help facilitate this rapid adaptation. However, these patterns are not limited to these traditional stress traits, nor to the couch potato gene. During my dissertation, I have also looked at the innate immune response, and I find that the same pattern persists. There's rapid and predictable change across seasonal time that repeats across years and locations, and that there are epistatic interactions among immune SNPs that vary with season that help pattern that response. So I've shown you that these populations under our noses are changing very rapidly. I would like to leave you with the idea that this could be happening in a very widespread manner for all of our organisms. So I'd just like to thank my collaborators and my PhD advisors, as well as the organizers, the Crow Committee for the honor of presenting in front of you, and the broader Drosophila research community.